Hello, it is time for Researchers Come. My name is Yoshinori Fukasawa. In this program, we introduce researchers from all over the world. Today's researcher is Dr. Akio Kanai.
Nice to meet you.
Nice to meet you. First of all, could you tell me what your current research theme is?
All right. I'm basically a molecular biologist and biochemist. But recently we combined also the technique of the bioinformatics. Using these kind of the technologies, we are currently analyzing the RNA molecule, not particular one molecule, but also the genome level or any kind of the RNA molecule, plus the RNA binding proteins. In the 21st century, a lot of the non-coding RNA is appeared in the database. I really want to know what kind of function are there, and we also get interest in the archaea bacterium, archaeas. It's living in such as a very hot spring, like 100 degrees. This the environment is really mimic for the Asian type of the Earth. That's why when I find some kind of the the regulation in the archaea bacterium, it must be related to the origin of the gene regulations. That's why I'm really interested in the RNA molecule and the archaea bacterium. Today, in this short interview, I focus on the tRNA molecule. This is one of the classical non-coding RNAs, where it is already known that tRNA involved in the protein translation. It's bind to the amino acid and move to the ribosome and translate it. And it's involved in the translation, but the molecule itself is the RNA. Plus, in some kind of the retrovirus, like HIV virus, tRNA works on the RNA primer for the DNA synthesis. That means that tRNA involved in every important steps of the central dogma: DNA synthesis, RNA, and translation. That's why when I find something new things in the tRNA, it must be related to the secret of the gene, gene regulation and the genetic codes. Recently, we developed a new software called Sprint, which is developed for the prediction of the tRNA identification. Since some tRNAs are still missing in the genome of the archaea bacterium. In these short topics, I'm going to focus on the tRNA molecule in the archaea. This is the figure of the tRNAs, and this is the intron-containing type of the tRNAs. Since they are well-known software like tRNA scan SE is recognize the structure of the tRNA without introns. The tRNA having a lot of the secondary structure, conserved structure, and this is the signal for the computational prediction. But when you inserted such a the intronic sequence in the tRNA, it's so hard to predict in the computationally. That's why our strategy is predict the intronic sequence first, because intronic sequence having some kind of secondary structures called helix, Barge helix, Barge helix structure, BHB structures in their secondary structure. That's why we predicted this structure first and removed this intronic station from the genome first. That means the in silico splicing, doing the in silico splicing. And after the splicing, we can easily identify the tRNA scan, tRNA molecules by using the tRNA scan SE the software. This is our strategy.
Could you point out what the most important part of this intron inside tRNA is?
Mostly, most important things is the structure of this Barge region, two Barge regions. Is that correct?
This region.
This region. This region is okay. Most important, the region is that this the Barge regions. It's a very specific secondary structure. And our software recognizes this structure and remove the intronic sequence from that genome. And this is a very good way to predict the intron-containing type of tRNAs. And recently we published very unique papers about the tRNA prediction and the whole set of the tRNA identification by using these techniques. And I'm really happy to introduce one of my excellent students, Mr. Junichi Sugahara. And he is the first observer of the paper.
I'm Junichi Sugahara, PhD student of Keio University. Nice to meet you. I'd like to introduce my research using splits. As Kanai-san said, the analysis of archival tRNA genes is being important to evaluate origin and evolution of tRNA molecule. However, many tRNA genes were still missing in several archaeal species. Therefore, we developed splits. This is the web interface of splits system. This software enabled to identify intron-containing tRNA genes, and by using this software, we conducted comprehensive screening of intron-containing tRNA genes from 47 archaeal species and identified total 2,100 tRNA genes, which succeeded to cover 99.9% of all codon tables in archaea.
It's almost perfect.
And previously, the content of intron-containing tRNA genes was estimated to be 10% of whole tRNA genes.
Do you mean this type of tRNA?
Yes.
Yep.
This type of tRNA gene is only about 10% of whole tRNA genes, and surprisingly, we observed marked enrichment of this type intron-containing tRNA genes. For example, in these 5 species, about 70% of tRNA gene candidates were found to be intron-containing tRNA genes, and half of which contained multiple introns, such as like this. This tRNA contains 3 introns at single molecule. And finally, all the results we registered SplitsDB. This is the SplitsDB. This is a novel and comprehensive web database of intron-containing tRNA genes in archaea. This is very user-friendly database implemented by CGI scripts. Our novel and comprehensive tRNA gene dataset, together with SplitsDB, will provide a new molecular basis for upcoming tRNA studies. Thank you.
Okay, sorry, I have a question.
Yeah.
You said this type of intron-containing tRNA, in this type of RNA, the number of intron is 3.
Yes.
Only 3? Or there's no intron
4 or 5?
4 or 2 or?
Yeah. 2 intron-containing tRNA genes were also found, but maximum 3.
Maximum 3.
Yeah.
Okay.
There is no 4 and 5 intron-containing tRNA genes.
The number of intron is 3 at maximum.
Yeah.
Okay, thank you. Thank you, J. Sugahara.
All right, thank you, Sugahara.
And before coming to this institution, I read your interview kind of web documents, and the document says you have a strong philosophy about research. As a scientist, you said you should. A researcher has a hypothesis to research, and then researcher shows or proves phenomena after making hypothesis.
Right. It's not my original philosophy, but when I was the graduate course in Tokyo U, my mentor, the Professor Shunji Natori, influenced me a lot. That means he very much focused on the originality of the science. That's why, as a professor, now I'm in the Keio University, I'm really, emphasis the originality is really important for the researches. As you know, especially in the molecular biological field, mostly the people just doing some kind of their routine works. When they got their genes, just they check their gene expression, that they check some distribution of the protein by the antibody or some kind of these stuffs. But most important thing is the originality in the science. That's why we first have some kind of the hypothesis first, and then check this hypothesis in some kind of materials. This is very basic way to doing research. That's why I really mentioned about in my homepage. That's it.
That's cool. As you guys might know, in modern biology, the experiment is getting routine and routine. So the impact of each research to me seems to be lower than before. As Dr. Kanai said, originality is the most important factor of research. Okay, it's cool. And secondly, could you tell me what you want to do in the future? As
Sugahara-kun talked about the tRNA molecules, and I myself interested in the archaeobacterium very much. But it's just materials. I really want to know what's the kind of Asian type of gene appeared in the early stage of the life, the beginning of the life. I'm really interested in the evolution of the gene and the evolution of the lives. That maybe next 10 years I will have some kind of the experimental work or the theoretical work on the origin of the lives, these stuffs.
You mean you want to focus on the origin of the life through your research?
Maybe origin of life is very difficult to doing something. But as I said, the archaeobacterium maybe the very still have some kind of the feature of the Asian type of the style of the. That's why when I find something new in the archaeobacterium, especially the focus on the RNA molecule, it must be related to the very early stage of the gene regulations. That's why my meaning of the Asian type of the regulation maybe maybe there. So I just dig out some kind of the early stage of the gene. That's my meaning.
Okay, thank you. I think the point of your research group is containing both informatic biology and experimental biology.
Exactly.
Do you have something special feeling or philosophy to comprise this team?
Yes, it's one of our institute aims that the exactly same student doing the bioinformatics, both bioinformatics and experimental biology, because when 20th centuries physics developed very much in their doing their both applied experimental physics and also the theoretical physics. But in the 21st century, bioinformatics is very powerful to doing the prediction or some kind of the simulation of these stuffs. It's another means that bioinformatics is have a kind of the value for the theoretical biologists. But theoretical biology itself is not, not sufficient. Experimental biological support is very much necessary for, doing so. That's why our institute focus on both, the bioinformatics and the experimental biology. And when you combine these, different techniques or different studies together, it must be a, very new finding in the 21st century.
Oh, it's interesting. You have a belief that our future scientist has some aspects of both bioinformatics and experimental biology.
Yes, that's
and right. To complete this belief, your institution is processing to educate students with both aspects.
Yes, that's right. We have started our graduate course almost 7-8 years ago, and we did when they are experiment student, when the student comes we teach the both programming and the lab techniques. That's the way
to educate students for future desirable students,
next generations,
and leading theoretical biology.
Hope so.
Thank you. And it's time to finish. And I think both Dr. Kanai and Mr. Sugahara showed you guys some interesting figures, and unfortunately it might be difficult for you guys to see these videos. We occasionally upload this figure maybe under this video file. Please check it out. Thank you.
Thank you. Bye bye.
Bye bye. Bye guys.